everyone. So today's talk is kind of paradoxically titled. I did that intentionally. So it's in cyclically mutating organisms, beneficial mutations are deleterious. All right. And so anyway, we'll unpack that as time goes on. The core of the talk is that I think some of the facts of biology have been misinterpreted, both by evolution and creation and intelligent design. I think that's and so my goal is to kind of paint another model, another way of looking at things, that might resolve some conflicts and conundrums, and get population genetics a little bit more in line with what's happening in actual mutations. So a little bit of background. I'm kind of painting this as a criticism of genetic entropy as a primary model of genome evolution. But first of all, I want to say that I'm not against the concepts in genetic entropy overall. I'm not against the concept of mutational errors. Our family's been horrendously affected by mutational errors, so I'm quite aware of them. I'm not against the general conception of genetic entropy, only the degree to which it is thought to operate. So there probably is some amount of genetic entropy going on. The problem that I'm finding is that I think more mutations are being included in that than should be, and that the models of mutation done by genetic entropy overly focus on problems in mutation when there are several things that happen in mutation which are non-problematic and possibly beneficial. What I like about genetic entropy is I think it's a fantastic model of an internally consistent neo-Darwinian outlook of biology. So basically, genetic entropy is what you get when you take neo-Darwinism and you make it obey its own rules. But I don't think it actually captures real biology quite as well, because I don't think real biology is quite as neo-Darwinian as genetic entropy takes it to be. So I think for a long time, I know that when I was growing up, I was taught that mutations occurred by happenstance, and I think a lot of us just kind of assumed that was correct, and we assumed that this was the way things really worked, and maybe we disagreed with the effects of that, but we didn't really think to question whether or not the core idea was true or not. So I think genetic entropy takes that core assumption and runs with it and shows where it goes, but I don't think the core assumption is necessarily true. So I want to talk about 3 questions that people often have and the common answers to them. Okay. So the first one is why is the mutation spectra that we observe in vivo dominated by slightly deleterious mutations? Okay, because if you take a sampling of all of the mutations that we know happen, generally they're dominated by slightly deleterious mutations. Second one is, what does this mean for long-term fitness? And the third one is the model being used Darwinian or intelligent design based? And so the typical answers to these, on the first one, most people say that since organisms aren't directing mutation, it is unlikely that they would find beneficial mutations by chance. Okay, so basically, they're dominated by slightly deleterious mutations because since these are accidental mutations, the chances of them hitting upon a better solution is very small. So what does that mean for long-term fitness? Well, selection is unable to remove slightly deleterious mutations because selection is actually somewhat of a weak force, especially compared to even just a randomized death in the environment. So for slightly deleterious mutations, selection actually is useless in removing them. And then most people think that this model is ID based because it shows that mutations can't account for evolution. Okay, now I'm going to find issue with all of these answers, but we will get to that. So I want to talk about two different models of spontaneous mutation. Okay, so first of all, I want to talk about the difference between spontaneous and adaptive mutagenesis. And so when I talk about spontaneous mutagenesis, I'm talking about mutations that occur that are not in response to any specific stressor. Okay, so they just occur. There's not something specific in the environment that's triggering them. There's not even like a stressful time for the organism. They just happen. Okay, spontaneous. And then adaptive mutagenesis is when a organism has stress, some sort of selective stress, and these are mutations that are generated in response, whether it's just a heightened mutation rate or there's specific mutations. So those are kind of the two modes of mutagenesis that I generally consider. So what we're going to talk about today is spontaneous mutagenesis. I've done a talk for this group on adaptive mutagenesis, which I've got a YouTube video of. I don't think we actually recorded the talk, but I recorded a similar one afterwards. And then I did a biocomplexity paper called Estimating Active Information in Biological Systems. So, so today we're going to cover spontaneous mutagenesis. So the typical model that most people think about for mutation is a linear model of accumulating mutations. Okay, so basically you have your genome and there's a mutation, so you get if your genome is A, then the next one's A prime, and the next mutation gives you A prime prime, then A prime prime prime, and so on and so forth. You're just linearly accumulating mutations, and so each one is different than the last one. So basically at each stage we're gaining mutations. They're rarely reverted. And that's just a probability issue, because there's just so many possible mutations that there are. So the chances that they're reverted is very low. You can essentially remove them from your model. They're so low. And so the fitness and then the fitness effects of these mutations are stable, roughly, relatively. So that's kind of the general model of mutation that we generally think of. And so, as I mentioned before, why we think mutations are mostly negative is that random changes to any complex structure lead to many more downward steps than upward steps. And why mutations accumulate: for a single adverse mutation, the chance of being set right by an inverse mutation is 10 to the negative 9th per generation, compared to 0.3 adverse mutations per chromosome set per generation. Makes that extremely small. And those are both from Hoyle's Mathematics of Evolution. So now the model that I'm presenting is going to be a cyclical model of mutation. Okay, and so this is a very different way of thinking about mutation. So the cyclical model mutation says that change is actually parameterized according to the design of the organism. Okay, so changes are not unlimited in scope. They're actually very limited in scope. There are parameters involved that an organism can change by. This allows design consistent changes to be more likely. And I have a paper on design consistent versus design inconsistent mutations that I'll reference at the end of the talk. So because these changes are parameterized, because there's only so many ways that they can go, reversions to previous good configurations are more likely. Okay, so in the linear accumulating model, the mutations were haphazard, and so the chances of reverting one are very small because the chances of any of them are occurring are extremely small. But in this, if you have your changes parameterized to certain types of events, it's a lot more likely that a reversion is going to happen. So the model kind of looks like this. If you have four, let's say we're going to deal almost entirely in toy models in this, just because that helps us think about it. So if you imagine, let's say that there's four possible configurations, and your organism starts in configuration A, then it mutates to configuration D, then it mutates to configuration B, and then mutates back to configuration A. All right, so here you have a cyclical model of mutation. So one thing that I want to point out that as I mentioned before, I'm not saying that all mutations are design consistent. Every part of biology has error in it. So mutation is not immune from this. Hearts have heart attacks. You get cancers wherever, and you also have and the mutation system, while I think it produces a lot of design consistent mutations, it can also break down as well. It also doesn't require that there's a fixed set of mutation, but having a fixed set of mutations is a lot easier to model. And I think it's a good first order approximation of how it works. So maybe a better term than just saying cyclical model would be a non-strict cyclical model. So, but I'll abbreviate it to cyclical model. So the way that I oftentimes help people think about this when they think about mutations, especially since we have been so focused on negative mutations, deleterious mutations for the last century, is that when we first learned about microorganisms, it was basically assumed that all microorganisms were bad. And the reason for that is simple, and that's because we knew about microorganisms because they were bad, because they were causing illness. And so it led to the belief that all of these microorganisms were bad and causing illness. And so what we should do is we should just get rid of all the microorganisms. But then it turned out that actually, something like 99% of microorganisms are actually good, and there's a very tiny number that are bad. And it just so happens that the ones that are bad are the ones that we're able to see clearly and medically at first, and we didn't recognize the other ones until later. And I think that's what's going to happen with mutations, is that on the whole, we're going to figure out that most of the mutations that actually occur are design consistent. And that there's a small number that are not design consistent, but they are way overshadowed by the design consistent ones. So I propose this really weird cyclical model of mutation. What is the evidence for it? Okay. So Wolf Eckhart Lönig, or Lönig, I think that's how you say his name, he came up with what he talked about the law of recurrent variation. And so in his paper, he references this guy. He says, during the years, so this is back when before they really did molecular biology, this is mostly talking about phenotypic mutation. So he said, during the years, each new large mutation trial showed that the number of really new mutations recognized for the first time was steadily diminishing, so that the majority of genetic mutations was already known. Then 20 years later, again, you have the larger the mutant collections are, the more difficult it is to extend them by new mutation types. Mutants preferentially arise that already exist. And then I think this is Lönig himself. He says, instead of the regular and perpetual formation of new useful culture varieties, subspecies, incessantly the same spectrum of mutants is reproduced, so that if, after a certain number of experiments, the method is hardly useful for plant breeding anymore. So the law of recurrent variation implies that genetically properly defined species have boundaries that cannot be abolished or transgressed by accidental mutations. So as I said, this is based on phenotypic variation. And even Lönig himself assumed that genetic variation itself proceeded in a Darwinian manner, but that it was more or less capped. The abilities were capped by the boundaries of the organism itself. However, simple sequence repeats are a very interesting class of mutations. So simple sequence repeat are basically repeats of short nucleotide sequences that just go over and over again, the same couple of nucleotides. Now the length may change. Some of them are single nucleotide repeats. Some of them are double, triple, quadruple, but just a short sequence that gets repeated over and over again. Now, what's interesting is that these simple sequence repeats have a much higher mutation rate than ordinary than the rest of the DNA. Okay, so the mutation rate here is 10 to the third to 10 to the negative second per base pair. All right, that's extremely mutable. Additionally, within these, the mutations that alter repeat number typically occur at rates orders of magnitude greater than single nucleotide point mutations. Okay, so what you have is that you have, when these simple sequence repeats, these are literally the most likely thing to mutate in an organism. Additionally, it mutates almost entirely by copy number variation. So in other words, we have this mechanism that very quickly can adjust parameters. So it's kind of a genetic way of having parameterized function. You just have links that are adjusting back and forth like tuning knobs. So you actually have a large plurality of mutations are in these repeat numbers, and they're very easily reversible. All right, so you have this, the thing that's most likely to mutate is reversible. They act as tuning knobs, and they've been shown to modify function in kind of general tuning knob-like manner. So anyway, it's very fascinating. So that's one area. You also have phase variable genes, and so these are basically discrete switches in the genome. And so these can rapidly turn biological systems on and off. So there's a lot of mechanisms for this. So SSR repeats, in addition to providing tuning knobs, can also do this on and off switching. So this especially occurs if the SSR is not at a the computer term is a word boundary, but it's not at a triplet. If it's offset, like if there's a 4 repeat, then adding another one can cause cause a frame shift out of frame. Yeah, so it can cause a frame shift, which can cause the gene not to transcribe properly, and so it will basically turn it off. So that's one of the mechanisms. Another mechanism is DNA inversion. So there's pieces of DNA that will literally turn themselves one way or another to turn themselves on and off. And then another one is DNA specific transposition. So you can have parts of DNA move around to turn things on and off. There's actually pseudogenes that get re-enabled, things like that. Then you have gene conversion, and this literally takes parts of genes and attaches them elsewhere. Okay, and so this basically turns on and off biological functionality like a switch. Okay. So let's see here. All right, so this provides kind of the raw material of an ID theory of spontaneous mutation. Okay, so you basically have pre-designed adaptations to different environments. They just get switched on and off. Or if you have something that needs to occur more or less, you can encode it in a tuning knob. So you have tuning knobs that adjust common parameters, and then spontaneous mutations occur to switch between adaptations or adjust the tuning knobs. Now remember, this is in absence of selection that this is happening. We'll get to that in a minute. And as they noted in the book, The Implicit Genome, which if you do nothing else, I suggest all of you go out and buy the book, The Implicit Genome. It's fantastic. It says, with 12 genes able to switch between on and off due to reversibly mutable repeat sequences, a population of bacteria descended from a single individual can explore 4,096 different versions of its genome without damage to other loci en route. Okay, so let that sink in. If you have a switch-based version of mutation, then you're able to explore space without damaging the DNA. Okay, so this is hugely important. Now we'll get, again, we've talked about the fact that most mutations are slightly deleterious, and we will get to that, I promise. But I just want this to sink in that this cyclical mutation model, which we have evidence is occurring in the genome, allows the exploration of different configurations that are design consistent, that are consistent with the biology of the organism itself. We don't have to hope that Darwinian mutations will accidentally find the right thing. The mutation system of the cell is already doing this for you. And then also from the implicit genome, the term implicit genome captures the additional information afforded by the predictable variations in nucleic acid content and organization. Okay, so basically you have the genome, but the genome also implies where it's likely to change. And you can use that to figure out how an organism is likely to adapt. All right, so now cyclical mutations in fitness. Okay, so the question arises, if mutations are designed, why are so many deleterious? And this, he wrote a paper called Fisher's Fundamental Theorem of Natural Selection Isn't Fundamental After All. And in this paper, he pointed out that there's two opposing definitions of fitness that are oftentimes conflated. The first one is the reproductive success definition of fitness. That's how many offspring a genetic configuration can produce. The second one is biophysical suitability definition of fitness. And this is how well suited the genetic configuration is for function. Right? These are two different conceptions of fitness that are not actually equivalent, although most people take them to be equivalent. Evolutionary biology takes these two things to be equivalent, but they're not. Now, if you have a lack of biophysical suitability, this loosely implies that you're going to have a problematic reproductive success. But the inverse of that is not the case. A lack of reproductive success does not mean that you're not biophysically suitable. So my contention is that in vivo spontaneous mutations are primarily a hedging tactic by the genome to maintain diversity. They adversely affect reproductive success, but they don't affect biophysical suitability. Okay, so we'll get into this a little deeper. So bet hedging. So these mutations are occurring spontaneously. So there's no active selection involved. So if I'm investing money, the idea of hedging is that I have my primary investment, which going on how the world, my present conception of the world works, my primary investment is what I think is going to make money. Hedging means, well, what happens if that thesis goes south? Is there something else that some other play that I can make such that if my primary investment goes south, this other investment, which I don't think is quite as good, will start performing well if my primary investment goes south. Okay, so that's what a hedge is from an investment standpoint. So now hedging limits your payoff, because what you're doing is you're taking money that you could invest in your primary investment and you're putting it somewhere else. Okay, so it limits the payoff, because if your primary investment just goes gangbusters, it would have been better to put all my money in there. But the reason why we do this is because it limits the downside. So hedging, it limits the payoff, but it limits the downside. So if everything goes bust, if I have proper hedges in place, I won't lose all my money, I'll just lose some of it. So the way to think about mutations in this sense is that if everything's going fine for the organism, it's still going to want to maintain some amount of diversity. It's going to want to diversify its portfolio in case something happens, in case there's some sort of environmental change in the future. Now, if the mutations were random in a Darwinian sense, this wouldn't work because the space of possibilities is so big that trying to find something that's successful in a future environment is not very reliable. So, so hedging doesn't actually make sense in a Darwinian sense because there's no way to that selection could find the alternate investment. There's just too many possibilities. The chances of having something that actually works properly is too small, given that the organism has no way of finding the environment and there's no feedback between the future of the organism and the mutations. However, since we have non-random mutations, we can have meaningful hedges because the genome is organized organized to generate functional alternatives. Okay, but here's the key part: these are by definition deleterious because if we use the reproductive definition, they're not going to be as successful as the current plan. They're hedges. Okay, so they are related to biological function, but since they are hedges, they are not as successful as the current configuration. So this is actually anti-Darwinian because the Darwinian mechanism, the mutations happen irrespective of organism function. Well, these happen very closely related to organism function. They are tied to the needs of the organism. They're just tied to the future needs of the organism. Now, there's been some literature on evolutionary bet hedging, but it's rarely been discussed in terms of a mutational strategy. And it's usually focused on traits where you kind of have identical genetics, but varying outcomes. It was more popular in the 80s, but there's kind of been a tradition maintained, but few of them actually deal with mutation as with hedging as a mutational strategy. So one other note on this is that if you add in stochasticity, a little bit of randomness within the parameterization, you get diversification without the overhead of communication. So mutations aren't random with respect to fitness, but they do appear to be stochastic. So this means that basically means there's a probability distribution. And if you determine hedges via a stochastic process, this means that a population can diversify without the overhead of communicating with other cells about which pathways to take. So for example, let's say that I have $1,000, okay, and I want to invest 10% in a hedge. All right. Now, as the owner of the $10,000, I can literally take 10% and move it over. But let's say that each dollar was autonomous. Okay. What I could do is I could just tell each dollar to roll the dice. They roll a 10-sided die, and if they roll a 1, they're the hedge, and if they roll anything else, they're not the hedge, and that will give you the same outcome. So they're each rolling dice, and so each one individually has a stochastic question of whether they're going to hedge or not. But that gives me kind of a fixed way of allowing all of the dollars to hedge, and they'll hedge the percentage I want them to. But I don't have to maintain coordination or communication among them to do that. So I want to give you an intuitive demonstration of this. So we're going to do cyclical mutations. So we're going to have 4 genomic configurations, A, B, C, and D. And we're going to have 3 different environments, E1, E2, and E3. And so we're going to have this environmental mapping. So in environment 1, A is going to double itself every generation. B is going to triple itself. C is just going to maintain itself, and D is going to double itself. In environmental 2, A is going to half itself every generation. B is going to quarter itself every generation. C is going to double itself each generation, and D is just going to maintain flat. And then environment 3, A is going to go flat, B is going to go flat, C is going to double, D is going to triple. Okay, so the A, B, C, and D are the genome configurations. E1, E2, E3 are going to be the environments. So we're going to start. Our starting point will be environment 1, and each genome configuration is going to have 100. Okay, so I'm going to leave the environmental mappings at the bottom right so you can remember what they are. So in generation 1, we've got 100 of each. Okay, so in generation 2, if you look at the mappings below, we're going to double A, we're going to triple B, C is going to maintain, D is going to double. We're going to do that again. We're going to do that again. We're going to do that again. Okay, so now which of these is the most fit? Well, the one that's most fit is obviously B. Okay, because it's tripling each generation. So by the reproductive definition of fitness, B is the most fit. So now let's say that we have a constant mutation rate. Okay, and let's just talk about per organism mutation rate. So whatever that mutation rate is, when we get to number 5, or generation 5, and so we're talking about cyclical mutations, so they're all going to just be mutating to each other. What is the most likely, if I were to choose a mutating organism at random, what is the most likely outcome? Well, to think about that, which organism is doing the best, has the most members? Well, B has the most members. So most likely, if I just choose a mutating cell at random, it's going to be a B. Okay, well then, is the mutation likely to be beneficial or deleterious? Well, I've got 3 things I can mutate into. All of them are less reproductively fit than B. So from a reproductive definition of fitness, the most likely outcome is that a mutation is going to be deleterious. Okay. So if a mutation occurs in generation 5, the most likely outcome is that it's going to be deleterious. All right. So environment switch. So now we're gonna have the same population starting where we ended the last time in generation 5, but we're gonna go to environment 2. Okay, so in environment 2 we have different population dynamics. Okay, so A is gonna half itself each generation. B went from being the most fit to the least fit. It's quartering itself each generation. C is doubling itself, and D is maintaining. All right, so here we're gonna go. So notice A is halving itself, B is quartering itself. B is just being decimated by this process. And so we're going to get to the end, and notice there's almost no B's left. Okay, they're almost all now. Where are they? They're all in C. So if a mutation occurs in generation 10, is it more likely to be beneficial or deleterious? Okay, well, in this case, most of the organisms are in C. And so most of the mutations then would be away from C. And so most of the mutations would be deleterious. All right, so now we're going to do an environment switch again. We're going to go for environment 3. And so we're going to do environment 3 for 5 generations. And so now we have D is outperforming everybody. So if a mutation occurs in generation 15, is it more likely to be beneficial or deleterious? Well, again, it's most likely to be deleterious because the most popular one is D. It's most popular because it's most fit. And so the mutations are going to be primarily in D, away from D. And so that's going to be, by definition, since D is the most fit, the results are going to be that they're deleterious. So you can see that even in these environments where all four configurations are designed to be fit in some environment, at any given moment, as long as they get to be consistently in an environment long enough, at some point, most of the mutations are actually going to be deleterious. So here we're going to go to the math. Okay, and so a little bit of a warning. This is early work. I did and this is also where I would like help. So if anyone has just genius ideas, please share them, because I would like to work this further. So a lot of this actually is based on kind of how Sal kind of was developing. He showed a proof for the fundamental theorem of natural selection, and this kind of starts with that and makes modifications. So let's start out with basic definitions. So you have basic population growth. The change in population over time is some rate times the population. Okay, basic population growth. Usually the rate is the natural log of fitness, but in our model, to simplify life, we're just going to say R is equal to fitness. Okay, so that will, because the natural log, so all we really care about is more or less fit. We don't really care about quantitative differences. So we're going to, so the log tracks greater than or less than. So we're not going to change anything by doing it. We're just going to simplify the math. All right, so P, we're going to split up our population into subpopulations. So every alternate configuration is going to have its own subpopulation. So therefore, the actual change in population over time is going to be the sum of all of our individual changes of population over time. So now we're going to do fitness-based growth. Okay, so the fitness of a given subpopulation is, so we're going to represent it as F sub I, and that's going to be the fit. So it's going to be the fitness function, which is going to take the environment at time t and the subpopulation I. So anyway, so that's going to give us a reformulated population growth equation. You're basically going to say the fitness. So it basically just says that R, instead of being a constant, it's going to be a variable. It's going to be based on the, which is based on the environment and the subpopulation. All right. So now mutations are going to have effect all the way through. So we can lose an organism from our subpopulation, whatever it is, to another subpopulation through a mutation rate. And for our purposes, we're going to assume that the mutation rate is constant, and we're going to measure it in organisms per time period. Okay, not per base pair, organisms per time period. So the change in population of a given population is going to be the general, the rate based on fitness minus the number that we lose to mutation. Okay. Now, on the other hand, other populations are going to lose organisms that, since it's cyclical, it's going to come back to our population. So we're going to gain, from other members of the population. So basically, so if we add the ones that we're gaining, so we're going to have the ones from growth. That's the first term. The second term is the ones we lose by mutation, and the third term is the number of that we gain from other people's mutations. Okay. So that last term can be simplified to that, and that's because if we look at all of the population that's not us, that's going to be the total population P minus our subset I. So so this tells us the growth in population of any growth of any subpopulation based on the environment and the relative fitness in that environment. All right. So the number of beneficial mutations is going to be basically, if you think about it, like if I'm mutating from A to B, or let's say I'm mutating from A, right? Well, whether that's going to be a beneficial or deleterious mutation is going to be based on whether or not that mutation went to a population that's growing faster or growing slower. Okay, so this is going to be basically, so the number of beneficial mutations is, we're going to go through each subpopulation, and we're going to count the mutations that are going to greater ones and going to lesser ones. So the beneficial, so that second term is the size of the number of configurations that are beneficial. So we're taking our mutations and multiplying it by beneficial ones. And likewise for deleterious, it's just the opposite. And so we can get a ratio of beneficial to deleterious by simply putting these over each other. And notice that since the mutation rate is constant, the mutation rate drops away, as well as that n-1 term. So basically, in short, you multiply each subpopulation times the number of worse configurations divided by each population times the number of better configurations, and that will give you the ratio. And I'd like to explore this further. That's kind of as far as I brought it. But notice that if the mutation rate and spread is uniform, then the deleterious rate is actually independent of the mutation rate. It's actually based more on population than the mutation rate. I don't know if that's interesting. Mutation rate is itself is variable, so if you don't model it as being constant, then all of a sudden that doesn't apply. So, but I noticed it was interesting. Some notes on this. Obviously, the model itself is highly limited. As I said, mutation rates are not always static. There's a lot more population dynamics. We're also only modeling path-independent mutations. So only if all of the mutations could go to all the other varieties. Okay, and but actually that's not the case. Some of them require more than one mutation to go from A to B or A to D. And so we're only modeling the ones that are path independent. And we're assuming that the distribution from each bucket to each bucket is identical, which is kind of another way of saying they're path independent. And we're also assuming that the environmental switch itself does not induce specific mutations or alter the mutational strategy. That being said, there's some things I think we can draw from this. I think that the model can be useful if we focus on non-induced mutations. We focus on things that have a static mutation rate, and there's a lot of things that do. If we focus on path-independent nearby mutations, so there are mutations that are relatively path-independent, and we can see those. Even if you find a single organism or circumstance which fits this model, it shows the limitations of Darwinian thinking about biology. So one thing that you can actually detect the phenomena even in absence of having an exact equation to match it. There's certain kind of markers that will help you determine this type of mutation. So that first one, I'd actually have to think about that first one some more, because that might be based on some additional assumptions that are not necessarily to be made. But the thing to point out is that the longer the population sits in a constant environment, the more the deleterious beneficial ratio will increase, right? And so basically that's what we noted in the example, is that as we leave it in the same environment, then it gets more and more dominated by the best configuration. But that means the rate that they're switching to deleterious ones is actually increasing. And then the other thing is that if you switch the environment, then it should cause a sudden decrease in the deleterious to beneficial mutation ratio. Okay, because all of a sudden the environmental mapping that was causing the ratio has changed. So you should get a sudden change in the beneficial to deleterious uh, ratio. And that's kind of what I think is most interesting, is to see if that happens in the lab. So some additional reading material. I did several papers on this historically, not getting into as many specifics as I did here, but some background reading. Talked about randomness and creation biology. Towards the creationary classification of mutations deals with the design consistent versus design inconsistent mutations. A little bit about bed hedging, but also, like, if you read nothing else, I hope you get a copy of Caporal's The Implicit Genome, and maybe read Sal's comments on Fisher's fundamental theorem.